Thanks very much. So this is going to be something that's a completely different laboratory than we're usually used to seeing. This is the International Space Station. It's an incredible laboratory. It's a fantastic place to live. I'm going to tell you just a little bit about what we're doing up there these days in terms of post-Earth, post-planetary, post-genomics. And then hopefully we can talk a little bit about Moon and Mars, because I've gotten pretty excited about that recently. So this is the first DNA sequencing in space. We had this idea a few years ago. Actually, the folks came to me a few years ago and they said, hey, we're going to send a sequencer to the space station. I said, that's a ridiculous idea. Why do you need to sequence DNA in space? You've got cold stowage. You've got a SpaceX capsule. Just land it back here. We're going to have a much better error rates, and you don't have to worry about mass, power, all the rest of those things. So I changed my mind a little bit about this in the last few years. The interesting thing about doing some of this analysis in situ is that we can actually start to get real-time results on orbit on space station. And so we had this great little setup. So this was my lab here, and you can see the space station is an interesting place. You stick your lab to the wall. So I stuck the procedure, the Surface Pro that was powering, and you can see right below that the very small nanopore sequencer. The post-it note is death warning to my crewmates that if they got near my experiment, I knew which module they were going to hang out in, and I didn't want any flying kicks to disrupt the 48-hour sequencing. So I got this all set up. Then, this is kind of a big experiment. You launch everything. It's got a lot of planning that goes behind it, and you have a few samples. If you screw this up, you don't get more samples from the planet. So I was getting ready to do this, and I thought, gosh, I've done a lot of pipetting in my life, but I really need to make sure that I do this exactly perfectly. So I went and got a pair of loupes so that I could magnify and make sure I had a good eyeball onto my pipetting here. Then I thought, I don't have enough light, so I went and got a headlamp so that I would make sure I had a really good visual field. And I said, well, this isn't actually enough, so I went and I got a work light so that I had some good task lighting in addition to all of the magnification and the illumination. The one thing I didn't consider is that actually when you're doing the pipetting, every force that you put of the pipette onto the microcell is going to have an equal and opposite reaction. So then I had to go get a handrail to secure my toes so that I would counteract the force of the pipetting. So this was a fair amount of experimental setup here. It's a lot of overhead, but you don't want your sample to go out the pipette and then and not go into the sequencer. So we did the sequencing. We did a mix of Lambda, E. coli, mouse genome. We reassembled it. This was very exciting. This got published. What's not published that's coming out soon, actually, hopefully, is that the next phase was then to use this for pathogen identification. So right now, the way that we do any kind of environmental microbiology on space stations, we take these swabs, we streak plates, we send these swabs. The astronauts package them up with a whole lot of tape. We put it into a Soyuz capsule. We send it back through the Earth's atmosphere. It gets very hot. There's lots of plasma. I did that as well. These plates land in the middle of Kazakhstan. We then put them on an airplane. We get them back to the Johnson Space Center in Houston. Then we do our clinical microbiology. This is sometimes a 3-month delay from when you actually are interested in the sample. So one of the things that we've been interested in is, can we actually take a swab and then determine right away, without having to go through this really long delay, if we have something growing in this machine that's your life support system. So we did this successfully. And there's now an entire interior microbiome of the station to sample. One of the reasons that we're also interested in space as an interesting place to interrogate human physiology is that everything you've ever thought about for fluids behaves completely differently. And since we are big bags of mostly fluids, this is a very interesting place to study things without a gravity field. So everything is upside down. What you think you know completely changes in space, and the properties of fluids completely change. So bubbles become a huge thing. Actually, there's a lack of the bubbles, the air going to the top of the tube, the fluid going to the bottom of the tube. So bubbles are all over the place in your scientific equipment. Another thing that's very interesting on board is what happens to fluids in the body. You're not subject to this gravity field. You don't have the fluid collecting in your leg. You have this massive fluid shift headward. you've got a lot of cardiovascular influences, some really interesting physiology that happens. You can also grow things in space in a different way, very delicate structures that would normally just settle to the bottom of the plate. We can try to approximate this a little bit by bioreactor-type spinning devices on Earth, but they have a constant fluid shear. You can't actually grow large structures. So since your cell cultures are entirely in free fall in this very large vessel, it's a very interesting place to look at things in the absence of gravity. So you can imagine all of the kinds of studies that you might do on this. As I said, there's a human physiology response to spaceflight. So it's everything from neurovestibular to cardiovascular. The bone loss has been popularized, the muscle loss as well. There's effects on the immune system. And then as we start to go really long duration and have humans be further away from the planet, we start thinking about things like nutrition, behavior, and deep space radiation effects. So as I said, this is a totally different lab, both externally and internally. Your lab is all four walls. This is inside the US lab of the space station. I had an argument with NASA for a long time before I flew because they said, well, we're getting ready to pack all of your things for spaceflight. And I said, great, I'd like 1,000 to 10,000 swabs. And they said, that's not available to pack on the spacecraft. This is going to be an upmass issue. I said, well, how many pants are you guys sending? You have one pair of pants per month and one shirt every week. I said, well, I don't need that many pairs of pants. Can we swap out the pants for the swabs? And I'll just go with less pants. And this went on for a while. They finally said, well, you can put the swabs in your psych support kit. And so I went up with 10 kilograms of pipettes and swabs because this is interesting psychological support for the astronauts on the weekend. So one of the things that the interior of the spacecraft is very interesting. One of the things I've been getting more interested in recently is the exterior of the spacecraft. And for a long time we said, well, there's microbiome on the outside of the spacecraft. This is ridiculous. And I'm not so sure anymore these days. I'm not sure that we're going to see a whole lot of viable organisms out there, but it certainly may be interesting to start doing some detection. Space is a harsh environment, but every time we say there's no way microbes could survive an environment, fill in the blank, we usually find them there. So this is Jeff Williams. This was out in a spacewalk that we did installing the International Docking Adapter. There are some challenges to sampling the exterior of the space station. So if you want to try this at home, get 2 pairs of oven mitts, go hang upside down from the monkey bars, and probably rent a sumo wrestler suit, and then attempt to do your experiment. So there's going to be there's logistical hurdles for exterior station microbiome, but I think it's probably a pretty interesting thing to look at. There's a lot of different materials that you see on the exterior of the space station. There's some really interesting things with, for example, thruster pluming or air vents on the outside of the space station, places that you might bring microbes or transport microbes to space. And I'm not completely ruling out the fact that we are going to see some really interesting things that potentially have been bought there by troposphere aerosols or that sort of thing. So it's a beautiful place to live. It's an incredible place. There's a lot of really interesting molecular biology to do there. We're also planning, I just came from a conference that's really looking at the science requirements for a lunar orbital platform. And I wasn't very interested in the moon for a while because as a biologist, you say, well, the moon is sterile, it's dead. It's extremely interesting geologically, but this is hard to get worked up with a lot of enthusiasm as a biologist. Until about 6 months ago when I started thinking, well, the moon is sterile, it's dead. This is an incredibly interesting test bed for future Mars exploration. So these great spacesuits that we were talking about were designed to protect the human from vacuum, but they were not designed to be sterile. So we leak microbes out the spacesuits. And anytime we're thinking about Mars, we're going to go to Mars, we're going to sample. Well, we're going to obviously bring our communities with us. And it's going to be a really interesting question of how we ensure that we do not go to a place with low biomass and find our own biomass. So I think actually lunar sampling is going to be a great place to test out some of those technologies. And maybe we could talk about that a little bit further. So.
All right. Excellent. Okay, Kate, well, let me first start off in asking you how you went from Whitehead to NASA.
Yes, so I was procrastinating on writing an R01. It's a true story. my friend called me up and said, well, there's astronaut applications online. And I said, well, I'm deep into specific aim 2, and there's probably something I'd rather be doing to distract me from it, so I applied to be an astronaut.
So for all those people that are having a struggle with R01 applications, it's something to think about, huh?
Yes.
All right, so you had a whole new perspective of, we're here, we're in about our day-to-day dealings on this planet, and you're looking at it from another world, and maybe you just can describe what that experience is like rotating around Earth and what it does to change your view of our world.
Yeah, I think it actually, first of all, it really changed my sense of scale. So I think, we, as biologists, we tend to think in the 0.22 micron to about 3 meter range. There's a lot of things when you're flying over the planet, you kind of have this innate human sense of time and distance, which gets really challenged by going at Mach 25. So it pushed me to think about, hey, you're coming up off the coast of Brazil, Europe's going to be in 7 minutes. You do this every day, and you kind of follow an orbital track. It goes over different places on the planet, and it goes at a speed that at the beginning seems ridiculous and almost unimaginable, and towards the end of spaceflight seems like this very natural thing. So if I look at a problem now, I just immediately add a couple of orders of magnitude on it. So I think that's one thing that spaceflight has done. The other thing that it's done has actually taught me to be very interested in life support and atmospheric conditions and air molecules. You don't think too much about the fact that you just have oxygen available to you all the time and your CO2 is scrubbed. This is just this very nice thing that happens on our planet. But I think it's, these types of photos, and you can see this is the aurora, but you can also see atmospheric oxygen. And all astronauts comment on this. You see the vastness of the planet and this tiny little shell of atmosphere. And you stop taking atmosphere for granted when we regenerate our oxygen by splitting water. We scrub our CO2. We constantly have high CO2 in the spacecraft. It's really this sort of interesting physiological thing to have humans living under that much high CO2. And you don't take for granted anymore the fact that you've got a very limited number of air molecules that are providing your life support.
It makes me wonder if you could do a virtual reality of being on the International Space Station. Do you think that could be a cure for all the people that question climate change?
I'm not sure. I'm not sure if this is a cure for climate change, but it certainly is. We suggest it fairly seriously for world leaders every now and then, because we do have this experience. I had a really interesting experience on board Space Station. I was up there for a long time. We had a launch delay. I was up there for a long time with a Japanese astronaut and a Russian cosmonaut, and the Chinese launched. A different space station, different orbit. I have never met these guys. I probably never will meet them. But the day they launched, I was so happy. I just floated through Space Station. I was so happy all day. And I went to dinner, and we're floating around at dinner, and I told these guys, I'm so happy today. And they said, we were so happy today, too. We just floated around all day, and we were so incredibly happy. We all couldn't figure out why. And my crewmate, Talk, said, it's because we've been 3 for a long time, and now we're 5.
Interesting. Now, the next thing, of course, you got into this, well, before I get into some of the medical sequelae, you touched on it, just about the day-to-day life of not having fresh water, how to sleep, how to deal with circadian rhythm, all these things. How did you cope with that for almost 4 months?
It's a steep learning curve. So you get up there, and I think it's probably like recovering from a stroke. You don't know how to locomote. You're not sure where your toothbrush is. The first day you get up there, I spent 20 minutes trying to find my toothbrush. It's the size of a 747, and NASA's got more things in space than you could possibly imagine. They all have a barcode. So you have to find your toothbrush. Then you find your toothbrush, you try to put water on your toothbrush. Well, the water's floating all over the cabin. This is kind of the story of every daily activity. And so there's actually this, you launch from the planet a really competent, capable person. You get to space, you can't feed yourself. It's a challenge to go to the bathroom, and you can't move around until you relearn all of the daily activities. once you hit that, though, there's learning that happens at about 2 weeks in flight, and then there's like a really deep understanding of how to function in a microgravity environment, in a 3D environment, that happens about 4 to 6 weeks into flight. And then once you do that, you're in homeostasis, and there's sort of a asymptotic, daily life and how to work and how to function up there.
And you have no problem sleeping?
No, it's actually great. There's no pressure points. So you just float. We are doing a fair amount with blue light and circadian regulation. We've actually changed out all the lights in the space station to be tunable. And you have to be careful. You can't go watch sunrise and sunset right before bed. There's 16 of them, and the Earth is very compelling, and you want to go look at it right before you go to sleep. But this is a bad idea for circadian. But if you manage to do that, sleeping is actually fantastic on board.
And was part of your training dealing with recycling your waste and all that stuff?
Yep, absolutely. And so your coffee that you drink in the morning becomes your coffee the next morning. It's a fantastic engineering problem, though. I mean, we're really looking at how do you support humans physiologically? How do you deal with waste? It's a 90% closed loop system on board. And we're thinking about things like, how do you make this a 100% closed loop system? How do you increase the reliability of the engineering so that you can have these systems that are effortlessly recycling human waste? This is definitely a problem that we could use help with in the developing world.
Now, with you being up there for almost 4 months, and supposedly people lose 1 to 2% of their bone mass each month, did you have that measured? Did you lose a lot of bone mass?
Yeah, so we do DEX measurements pre and post flight, and I actually lost very little bone mass. What we've switched to now these days is a resistive exercise program. So it requires a lot of time investment. It's 2.5 hours every day of resistive exercise and treadmill running or biking, and that basically gets you bone maintenance. You maintain most of your major muscle groups, but we're now finding that we have a lot of paraspinal muscle loss and small muscle groups. So a lot of the stabilizer muscles are rapidly degrading. That's something that is not so much an issue. If you lose that, you just go ahead and gain that back. You really don't want to have any of the trabecular bone loss, because when you gain that back, it's cortical only, and you change your tensile strength. So you absolutely don't want to lose any bone, but you can afford to lose some muscle and then get back later.
You touched on the fact that there's such redistribution of fluids in the body, and so some people have experienced, visual loss, retinal edema, cognitive hits. In fact, when the ISS was here a year and a half ago, I talked to a couple of astronauts who'd had cognitive hits. Is that something that that you worry about? You think that's idiosyncratic, or could it be predicted? What are your thoughts?
So the papilledema and a lot of the eye issues, we certainly have been able to observe it, and we cannot, right now we do not have a very good predictor. Some people have zero vision issues. Some people have optic disc swelling. We've got cotton wool spots, papilledema, vision changes, permanent vision changes, and it doesn't seem to be something that we can currently predict. There's a lot of thinking going on right now about what could be driving this? Is this intracranial pressure? Is this, fluid shifts moving around? Is this high level of CO2 that we're constantly experiencing on board? And what is it that makes some astronauts predisposed to this and others not have any symptoms for the same duration on the same mission in the same environment? And
your colleagues, none of them had any of these?
So I don't know specifically whether or not they had any of those things. I do know that so far it seems to be segregating. The females don't have it. I didn't have any vision issues.
Interesting. Wow. That's one of the things I was most curious about, is because if we're thinking about going to Mars someday, which supposedly now it takes about 6 months to get there and back, another 6 months. You're talking about some major stress on the body. You'd like to predict who's going to be able to make the trip. And the question is, should we be sequencing, or is it just female gender? That's the answer. Should we find out who deals with the stress? Because you in that one slide, it means every body, every part of the body, every system is stressed. And it's not even just accelerated aging. It's even the pathogens are made worse. Immunity. I mean, there's not one part of the body. cardiac muscle atrophy, the whole works. So
Yes.
who can tolerate that? That's the question.
Well, and the question is also, you can tolerate anything for a while. And part of our training is you go out to the Navy and they put you in a helicopter upside down and drown you a few times. I mean, you're trained to tolerate a lot of stress, and you can recover from anything. You're going to come back and you're going to have a rehab period. What we don't want to do is have something where somebody's going to have a problem where they would need any kind of definitive medical care, because that is going to be very hard to provide. Right now, our answer for most issues is to do an emergency undock from space station, and so this is how you do a lot of the risk mitigation. So probably there's going to be a lot more risk acceptance, but you certainly might do a fair amount of screening and trying to determine who's predisposed to things. How are we going to select our astronauts for a long-duration Mars journey is, I think, an extremely interesting question that's going to start coming up for NASA.
I mean, just getting kidney stones in space.
Absolutely.
That's pretty common, especially in men.
Yeah, so we do have it's about a 200-page volume medical certification requirement, and we have the world's most intensive physical, I think, for your space flight. You have to receive an International Space Medicine Board certification. And so we do a lot of screening for those kinds of things, but it's screening with current diagnostic assays. We actually don't use any kind of genomic screening at this point.
So what is the prospects for timing of going to Mars, and would you like to go on that mission?
So, yes, we have these debates all the time, and it's one of the great, sitting around in the astronaut office drinking some beers after work. Oh, would you go to Mars? Go. And an hour later, we're still talking about it. So I've kind of decided, yes, if I have enough interesting things on the iPad, that this is going to be okay.
You can really load it up.
Yes,
huh?
you got to really load it up. You're going to be in a very small capsule. If you think the space station is a confined environment, we're going to have a lot smaller environment for a Mars trip. I think we're going to do a lot probably with this lunar orbit, where we are getting farther from Earth. It's a bit of more of an engineering challenge. You don't have this real rapid return. We can undock the Soyuz and land anywhere on the planet in not that many hours. You undock, you burn your deorbit engine, and you land 33 minutes later. Now, in an urgent kind of fire or depress situation, if you really have a problem with your vehicle, you can land anywhere on the planet, this is why we do a lot of survival training, but you can still get back to Earth very quickly. So as we push on to a lunar distance, now we're talking a 3-day, a 5-day, depending on your trajectory, type return. Then you can do longer, year-long checkout missions, and then you're going to eventually move on to a longer Mars trip, where you really, once you fire your engine, you're not coming back. You're going to deal with anything that comes along.
And if you were to guess when that might occur, the manned trip to Mars, when do you think that would be?
2030s is,
really?
I
Not
think
that far off,
yes,
really.
I think it's the kind of thing that we're starting to solve a lot of the engineering issues. We have a lot more questions to answer, but it's coming up right around the corner.
Well, that's your whole pioneering effort in many respects, but also of sequencing, and the fact that you could use a MinION and you could do something like that in Mars. That's going to be, you could never do that. Bring the stuff back. You can get the answer right in real time, right?
Yeah, and so this is one of the things that we're thinking about is how, this is going to be one of the questions for the lunar orbits is, what do you bring home, and what do you really start to push to do in situ sequencing? Now that equation, everything that you launch from the planet and put onto a space station or an orbiting craft has to be balanced for mass. It's the mass to launch, it's the power requirement, it's the data requirement, it's the sample processing volume and the crew time. And so you start to balance all of that against sample return, and in some cases it comes out that it's better to launch very small, portable, robust pieces of equipment and do the analysis in situ and return just the data. So we think about these kinds of things as well in remote medical environments. There's this kind of first-order approximation. Anything that I could put in a backpack or the back of a Land Rover and bring out to the middle of Congo would probably be a good space instrument. And so there's a fair amount of parallels for how you deploy things in a really resource-limited remote setting that are all the kinds of things we think about for spacecraft.
And the last question I had before opening up for a few others is that, what your thoughts are of the efforts going on right now, for example, with Elon Musk and the Falcon efforts and these other unmanned but very, I think, intense efforts to try to push this whole effort forward.
Yeah, it's incredibly exciting to see this, and we have so we have cargo vehicles that on space station. I together with my crewmates grappled a SpaceX, docked it to the space station, unpacked it, filled it full of samples, and sent it back to the Earth after a month. So we have orbital cargo vehicles as well. We've got a lot of companies that are working towards being commercial providers to launch humans as well. So this isn't just these aren't just cargo vehicle efforts. This is really the NASA backbone for our space infrastructure to the space station, as well as to eventual lunar platforms and beyond. So it's to me these are really exciting efforts. I like to see the progress that we're making, and I'm really looking forward to going down to the Cape and watching some humans launch off of a vehicle here. So this is coming soon.
Is there any dismay that this is not all part of NASA, that it's now this all private stuff, or doesn't really matter?
Well, no, it is, but it is part of NASA. I mean, these are going to the space station. This is how your swabs or your pants or whatever cargo you've argued for is actually getting up to the space station.
So it's really integrated.
Yes.
Yeah. A couple questions before we wrap up.
Thank you for coming here. And last time I heard you talk in person was at Whitehead. And at the time, I think you were doing research on smallpox, and you showed pictures of the hazmat suit you were wearing. And the
different kind of
subtext on that was your mother wasn't very happy about that.
Yeah. She's a little less happy about me going to space.
She could deal with the hazmat all right.
Yes.
Compared to this. Huh?
Yeah.
Wow. Anyone else? Well, you're amazing. That's probably the best way I could sum it up. We're so grateful for you to come here and enlighten us about your experience, and we wish you well, and you and all your colleagues in your continued efforts.
Great.
Thanks so
Thank
much,
you.
Kate.